Started.
We have 63 people on.
Yeah, the people will keep coming in. Yeah.
Okay.
So welcome everybody to our ASA Biopharmaceutical section webinar series. And today we're going to continue another with our mini series of partnership with statisticians and clinical professionals in drug development. Today we have the honor to have two prominent speakers for our webinar, who are Dr. Jay Herson and Dr. Michael Ewer. Dr. Herson received a PhD in biostatistics from the Johns Hopkins University. He has had a long career in oncology trials, which included faculty appointment at the University of Texas MD Anderson Center, and eventually he has his own CRO, Applied Logic Associates, which became part of WestEd in 2001. His primary interest is statistical applications in drug safety and is author of Data and Safety Monitoring Committees in Clinical Trials. Jay has served as Senior Associate in Biostatistics at the Johns Hopkins Bloomberg School of Public Health since 2004. Dr. Ewer received the MD from the University of Basel, JD from University of Houston, and PhD in public health from the University of Texas School of Public Health at Houston. He joined the MD Anderson Cancer Center faculty as a cardiologist in 1978, and he pioneered in the study of cardiovascular adverse events in cancer treatments and is still a thorough leader. Michael also teaches health law course at the University of Houston Law Center. So Houston has been an important part of both authors' career, and I'll turn the floor to Jay and Michael because they have a lot to share today.
Okay, thank you for that introduction, Vivian. I assume everyone can hear me now. We are going to be talking, as Vivian said, about real-world data, especially for cardiovascular adverse events in oncology clinical trials. And although Houston has been a part of Michael and my career, you would think that's where we met, but that is not where we met. We met, one second. The slide's not advanced. Hold it. Yeah, that's right. All right, where did we meet? We met at PS 173 in Queens, 4th grade. That's 9 years old. For us, this was before Queen Elizabeth and before President Eisenhower. And we did overlap at MD Anderson, but we never met there. I heard Michael give a talk at an ASCO meeting in 2006. I said, could this possibly be the same one who stood in front of me in 4th grade? And it was, and we've been collaborating ever since. In 2022, at the beginning of this year, Michael said he and many others are interested in the cardiac adverse events associated with the anti-cancer therapy, but concerned about the discrepancy between the reported frequency of certain cardiac adverse events seen in the post-market published reports based on sporadic report databases. And it's the sporadic report databases of adverse events, that's what we're calling real-world data today. And the difference between those reports and the pre-market frequencies of similar events based on the randomized controlled trials, the RCTs. The clinical guidelines for safety are being developed based on sporadic reports literature, because the sporadic reports literature are current and the RCTs were some time ago. And we sought to look at these differences and provide a perspective. And Michael is going to talk about possible problems about data from sporadic reports getting into guidelines. We shouldn't forget that pre-market and
Could I turn on my, speaker?
hello?
Was that? Yeah.
Please put yourself on mute.
We shouldn't forget that pre-market and post-market activities are aimed at establishing risk-benefit. Risk-benefit, not just risk. We cannot balance without considering efficacy. But how good are the tools for assessing safety anyway? If these tools are imperfect, how can we separate signal from noise?
Jay, are you telling us that if we put junk in, we'll get junk out?
Yes, yes, that is exactly what I'm saying, my fourth grade friend. And I have a lot more, we have a lot more to say. So in reviewing the RCT and the real world data in the literature, practicing oncologists can be confused by the discrepancy in cardiac AE frequency, and we're going to try to help you understand those differences today. Patients may be denied effective therapy for fear of an adverse event, but we are not addressing efficacy data or formal observational studies in this talk. Observational studies are also real-world data. We're not talking about that. We're talking about post-market communications based on sporadic reports. Those would be from insurance claims and other databases, and I'll describe those later. And these databases attempt to further characterize cardiac adverse events. So as an overview today, we're going to talk, Michael is going to run us through the cardiac AEs and cancer treatment. And we'll talk about the sporadic safety reports, their sources and uses, and they do have important uses, as some of you may be thinking right now. Randomized clinical trials, their strengths and limitations. Real world data, yes, they have strengths and also limitations. And what are the attempts to analyze real world data and statistical and clinical concerns on that? And real world data, there are prospects for the future. And I'm going to go over that at the end of this talk. And then we're going to have final words, both clinical and statistical. And now Michael is going to run us through the AEs.
Okay. First of all, thank you so much for the organizers for the invitation. It's a great pleasure to be here. And moving right along with this, real cardiovascular events in cancer treatments, and sometimes some of these are considered to be secondary or not terribly important. So I thought it would be interesting for this group to very briefly kind of put them in a context so that we wouldn't be leaving anything out. From the cardiac standpoint, and we can separate cardiac and vascular, from the cardiac standpoint, we have dysrhythmias, and we look at electrocardiograms, and we look at clinical correlation, syncopal episodes, and things like that. We look at myocardial dysfunction. Myocardial dysfunction at its extreme can be congestive heart failure and even death. Myocardial dysfunction can be permanent, caused by cell death. It can perhaps be transient due to a variety of things, but we think about myocardial dysfunction as well. We also think about pericardial problems, and pericardial problems are most commonly in the oncology setting the result of radiation, but they can also be the result of chemotherapy interventions, and we can have acute pericarditis, we can have pericardial effusions that may lead to tamponade and require interventions, and we can also have constriction that's a very serious problem and sometimes amenable to surgery. So on the cardiac side, we have dysrhythmia, myocardial dysfunction, and pericardial problems. But we shouldn't forget that cardiovascular also includes vascular, and vascular can be hypertension, hypotension, and thromboembolic events. All of these occur in cancer patients separate from the treatments that they are given, and also occasionally due to the treatments. And it's our job as oncocardiologists, if you will, to be able to separate this so that the interventions are appropriate. When we think about adverse events in cancer treatment, we have to ask ourselves, are they related? Could they be related to a direct or an indirect toxicity or reaction to the drug in question? When we say direct, some drugs are specifically toxic for the heart. We can think about the anthracyclines that result in cell death. But we have other things that can put strain on the heart. We have drugs that cause an increase in blood pressure, and those drugs will secondarily affect the heart. So many clinicians will assume that something that we see in the cancer patient is actually caused by the drug we are given, because we're giving something and then something bad happens. But cause is not always related to effect. And so we ask ourselves, what is causing this? Are they true events? Could other factors be the cause of the reported events, or could they be false positives? So we have two other possibilities. There are the real events, and the real events that are not directly related to the agent of interest. So what could cause something that could be confused with an adverse drug reaction, but isn't really related to the drug? It's related to tumor progression. It could be related to metabolic or endocrine changes. It could be related to random occurrences, also seen in the general population. Or these things could be, and this is what Jay and I have worked so hard to drive home, could they be a surveillance artifact? And a surveillance artifact is a finding that deviates from a true value and is the consequence of imperfect measurement tools. We need to separate the surveillance artifacts that are real because something is wrong in what we're doing. So we see an abnormal number. Or could they be a true event, or could they be an event that is real but unrelated? So kind of think of that as we do as clinicians, and we think of false positives as a test result indicating a problem that either is not present or is present but is caused by something other than what we're interested in. So this is kind of where we stand. Surveillance artifacts or other false positives may be interpreted as adverse events. And when the clinicians get it wrong, it leads to clinical decisions that are costly, maybe dangerous, especially if misleading to incorrect information, such as events become part of clinical guidelines. Good grief, do we ever have guidelines that are over-inclusive? But thinking that a false positive result is a true cardiac event may result in the interruption of meaningful therapy. We thought you had a cardiac event, we stopped this drug. Now you have tumor progression. We're sorry about that, but we think we did the right thing, but maybe we didn't do the right thing. False positives and false negatives reports may be underappreciated as well as overappreciated. And so Jay and I attempted to quantify the extent of false positive results that occur with at least one of the things that we use to look at cardiac events, and that is left ventricular ejection fraction. And so this came out in the paper that you see there, false positive cardiotoxicity events in cancer-related clinical trials, risks related to imperfect, non-invasive parameters. It's interesting to note that we had considerable difficulties in getting enough attention to get this, which we both feel is of fundamental importance, actually published. Errors are propagated in multiple measurements. So if you have a false positive, and if your test has a small percentage of false positives, and you do it a lot of times, you will add the likelihood of false positives. The more often you do the test, the more likely you are to find a false positive test if the mechanisms, the parameters that you're using are imperfect. And this is especially important in cases of ejection fraction. So we looked at this in a number of trials, and I don't think it's necessary to go through these individual trials with you, but these are fundamentally important trials in breast cancer. The breast oncology thinks of these terms that are over on the left side of your image almost as household terms. They go around and they talk about NSP, ABP, B31 and things of this type. What is important, though, is that these studies reported left ventricular ejection fractions declined, and they're all kind of in a pretty narrow range. The ones in the light blue shade are the ones that fell within the range of our false positives. So we really need to ask ourselves, is much of what is going into the literature now as cardiotoxicity to some extent or even to a greater extent based on false positives? The blue highlighted ones fit the model that we presented. And Jay's going to give you a little bit more insight into the statistics of that.
We did a computer simulation of thousands of patients, and there were two sources of variation. One was clinical source, and the other was errors in measurement of the echo instrument itself. And we found that the false positives would be at 3.6% with a narrow variance. And so what Michael is pointing out now is, if you look at the 95% confidence interval, 3.6% falls within it. Now you have some, you have the 9.4 and the 8.6 in those two important trials in second column, but those are where they measured they measured LVEF either 7 times or at least 9 times. The more times it's measured, the more likely it is to get a false positive. And we didn't have in our simulation as many as 7 or 9 times. Now, if you go to a sporadic report database, there was LVEF decline, and that's reported once, and we don't know how many times they measured that in this patient before this got into an insurance claim. And then the next time the patient comes in, oh, LVEF is better again. Wow, I intervened and I have returned this patient to normal, whereas it might have only been a false positive in the first place. So I mentioned the sources of these sporadic data. The FDA has one, FAERS. CDC has one for vaccines, VAERS. And Vigilance is similar for Europe. Of course, you have insurance claims databases of the big insurers, and Medicare is one of them. There's smartphone reporting these days. We're getting data from wearable devices and social media. People are doing natural language processing, going through social media and coming up with keywords to see what are patients complaining about. We're not going to deal with that issue today, mostly insurance claims and the FDA and the CDC. Now, there are many important uses of these sporadic safety data. Pharmacovigilance is an important post-market activity in the pharma industry, as many of you know. And then public health surveillance and the CDC. Early signals, they alert to rare events not observed in the pre-market RCTs, and we have to look into these. And FDA takes action. They can remove from market, black box warning, make dosage changes, or make a post-market requirement, a new post-market requirement. Again, FDA, it can be used at FDA in a positive way for a label extension. We can study drug-drug interactions as a paroxetine and pravastatin came to mind through these kind of databases. Long-term effects, we don't get the long-term effects from just an RCT, and we can use the data to design new trials. And usually, unfortunately, the data can come into court. A plaintiff's lawyer can use the data to show, of course, my patient had this adverse event. Just as an example from CDC, their weekly reports, this comes out of their database. We learned that COVID-19 was safe for children. And unintentional overdose reports are part of CDC's database. And of course, CDC gave us the running tracking of the opioid problems. And I hear from reports to FDA, the felbamate, after 100,000 patients were treated, we started to see cases of aplastic anemia, which would not have been observed in the limited clinical program. I might say also, before we get into RCT data, this is the 60th anniversary of the thalidomide and the thalidomide DES, and we learned all of that from sporadic reports as they existed 60 years ago. So now we're going to go into the RCT data, clinical trials, the strengths and limitations. Well, as a statistician, naturally, I will cover the strengths. It's considered a gold standard. Treatment assignment is by randomization. That is not unimportant. We get data that have been cleaned and cleaned and cleaned, and data on concomitant meds, comorbidities, and important covariates. There's a standardized timing and measurement of cardiovascular function, and the events are adjudicated by committees. And we have data on compliance, exposure data, and also we have data on both risk and benefit. Is someone have to mute?
Yeah, well, 1 second, Jay. Lakshmi, you're not on mute, and we hear you talking. Please put yourself on mute.
So, and we have data on both risk and benefit. So as I go through these, think to yourself, do we get all of this or most of this or any of this? And in what form do we get it from the real world sporadic data? So there are limitations of RCT data. First of all, it's a construct for regulatory approval. The patients are not representative of those seen in practice. There's a narrow eligibility, and we have to stick to inclusion and exclusion criteria. There's a short follow-up time, and there may be in the trial even early termination for efficacy, but then we have even less follow-up time. They may exclude cardiac vulnerable patients. For example, those with an LVEF less than 55 may not have been eligible for the trials, but they'll receive the drug post-market. And as we've said, it's not useful in detecting rare events. And AE frequencies can be underestimated due to splitting between the different measure preferred terms. Now, we do have the SMQs now, the standardized measure queries, and FDA has their own queries, and companies in industry have their own proprietary queries. So we're doing better on this, but then again, different groups use still use different queries, but it's better than it used to be. Now, Michael will take over real world data, strengths and limitations.
Thank you, Jay. Well, if we're talking about real world data, we have a broader population of patients. And this is important because if we are looking in a clinical trial to define a particular problem, and so we exclude patients that may have pre-existing conditions that might be confounding factors. When we look at the broader population of patients, then some of those will be included. And so risk factors may play a role. And another real world data reality is that if something is very, very rare, then it might not get picked up in a clinical trial of 100 or 200 patients. And the idea of a possible rare event might get picked up in real world data where it couldn't get picked up. So if the likelihood, let's say, of developing a myocardial infarction that happens in 1 out of 3,000 patients in the general population, and all of a sudden it's 3 times as great, and we see it in 1 in 1,000 if somebody is taking a particular drug. These are serious events that may be rare, and so they can be picked up. And it's important to have input if we're going to be looking at some of the newer techniques of precision medicine that simply could not be covered in random clinical trials, and we may be able to pick this up in real world data. So real world data is important. Sometimes, though, real world data is based on de-identified information. HIPAA requires 18 things to be removed from data so that it can be de-identified. But then if you don't know things that are important about that individual, like date of birth and things of this type, it may be impossible really to use that in the ideal way. So a person's name or record number may not be important in tabulating the extent of a problem, but if you want to go back and look at the record on a person, you may not be able to find it. So important variables could be lost in the de-identification. Treatment assignment is not randomized. So we have no data on severity. We have no data on the duration or whether or not the patient had benefit. All of those things are picked up in clinical trial data. We have no data on exposure to drug, especially when they're variables. So for databases, underreporting of well-established adverse events is certainly possible. Transient hypertension might not be picked up. And so we have things that are missed in the real world data, and we have things that might be included in the real world data that are not real events. We also have the possibility of double reporting. So we have people that may be in database A and database B, and there's no way to exclude duplication. And so the reported incidence may be artifactually increased. And then there's always concern about Medicare reporting. Medicare reporting can be in some ways based on billing and other factors.
Yeah, Michael, may I just add on this? New York Times reported recently that the large insurers, in order to get more money out of Medicare, they are, if a patient had a cardiac adverse event 4 months ago, they could put a claim today for another cardiac adverse event of the same type, And to Medicare, they would say, oh, the poor patient got it again. Now that will not go into the patient's EHR. That is only for Medicare, so they can bill Medicare again. Again, this is a developing story, as they say in journalism, but this is what they found. Now, if this is as widespread as the New York Times found it, someone's going to be analyzing these data sometime and have more than one adverse event when a patient had only one.
I'm not sure, Jay, are you talking about greed, or are you talking about fraud, or are you talking about both?
Both.
Okay, I just wanted to put that in perspective, because I think the New York Times article did make it clear that these were not legitimate variations. These were, in some respects, incorrect or fraudulent claims. If we think about the limitations of real-world data, claims data may be submitted to achieve payment for services. And this is right along with what Jay was saying. Whether we like the situation or not, we have situations where billing becomes the main focus in some instances. And how do we make sure that we are billing in such a way to not lose potential revenue is the concept. And sometimes billing of certain types of events, cardiac events, can get stretched. An example of how that might get stretched is if somebody is getting a cardiac ultrasound for let's say post-cancer treatment surveillance, but they're really doing pretty well, but it's important that they have surveillance according to the guidelines every 6 months or every year. That sometimes gets put down on a code as congestive heart failure because that's what they're looking for, but not something that they actually find. So different providers use different codes, and that can be a problem. No data on comorbidities or concomitant medications is concerned. And that can also play a role, because if somebody develops a problem that could be related to drug A, or could be related to drug B, or could be in some way related to both, it may not be possible to separate those comorbidities. And even deaths may be a problem there, because they may not be reported because you can't bill for somebody, for something that you've done on somebody who is deceased. When we think about that, and electrocardiograms are listed here, but probably more commonly are cardiac ultrasounds. And the frequency of cardiac ultrasounds may be much, much less in random controlled trials than in real world data. And if you do more tests and they are imperfect, you will get more false positive or false negative results. If we're thinking about adjudication, cardiac events are not adjudicated in real world data because it's simply impossible to do so. Cardiac events may have occurred independently of treatment. Cardiac events are common in the general population, and they may be more common or equally common in the cancer population in some instances. And how do we separate that? And it's sometimes very difficult to separate it. No information on how we measure the ejection fraction is often made. Sometimes the echocardiogram is technically problematic. Sometimes it uses enhancement. And so these things are not totally perfect. And patients may change insurance carriers when they change jobs, and that may cause a gap in reporting, and the frequency of surveillance is less unified than in random clinical trials. There's no adjudication of the cardiac event, and so we have all kinds of problems. In an ideal world, now let's take a deep breath here for just a second. In an ideal world, the limits of random controlled trial data will become less relevant as clinical data from real world supplemented. If the real world data were perfect, we might start with data from the randomized clinical trial, and that would evolve over time as we had really good real world data. And after a varying period of time, depending on the nature of the events, real world data should emerge as the gold standard. But ladies and gentlemen, we are not living in an ideal world when it comes to the surveillance of events in cancer patients and in other areas of medical care. And I turn it back to Jay.
Thank you. Now I want to cover analyzing real-world data. How do we analyze it? Well, first let me say, not to put some more nails in the coffin, but there's no denominator data in the real-world data. If the patient did not have an AE, we would not know of their existence. That is what's sometimes called informative presence. RCT data are global, but claims data are only the US. And there's an incompleteness and inaccuracy. And there's neither risk nor benefit, just isolated AE reports. And again, as we've said, no information on dose and frequency and or on the concomitant drugs. Now, analyzing the data, disproportionality is the main way that epidemiologists have attempted to analyze these data in the past. It's a proportion of, they call it the proportional reporting ratio. It's a proportion of an AE type on a specific drug divided by that proportion of all other drugs that happen to be in that database. Now think of that. I mean, the specific for AE type specific drug, ejection fraction decline, a specific drug. And then look at that same proportion on all other drugs that happen to be in the database, but the patients on those other drugs are in that database only because they had an AE of some kind, maybe not cardiac, but some kind, or they wouldn't be in this database. So there is a similarity that came out a few years later called the reporting odds ratio. And if you know how to change a proportion to an odds ratio, as we do in epidemiology, it's a direct analogy, and it has all of the same shortcomings. And it has, these two have been severely criticized as a measure of risk. Now, if you ever want to read a report based on sporadic data, this example of cardiotoxicity in the TKIs for CML leukemia. Tyrosine kinase inhibitors is TKI. These people did an incredible job, and the paper was published in Cancers. They went through 20 million FAERS reports, which included 1.3 million anti-cancer drugs. And then they looked only at the TKIs among those drugs, risk, that's disproportionality of cardiac failure, and a 3.8-fold increased risk disproportionality of ischemic heart disease.
Jay, stop for just a second. If you wanted to go through 20 million charts and do it correctly,
I don't have anything. So automatically English.
Somebody's not on mute. But if you wanted to look through 20 million charts, you have any idea how long that would take?
It would take me longer than the average person, I think, but how long do you think it would take?
It would take 65 people their entire career. I just did the math for the fun of it as you put up 20 million. This is why we don't have really good review of these records. 20 million records is a lot of records. Sorry for the interruption, but I just thought, you know, wait a minute, what are we missing here? Why don't we go through those 20 million charts? We need 65 people to spend their entire 40-year career doing it to review all of those records. Go ahead, please.
Yeah, and when the humans review the charts, they know what to look for. They know the limitations. This is just computer records where they can go through 20 million reports, perhaps in a half hour by computer. Now, then they looked at cardiac failure among, that is just one type of cardiac adverse event, cardiac failure among the TKIs for two drugs that were the leading ones. Dasatinib, a 4.1 disproportionality, there were 363 cases, and bosutinib, 3.5 disproportionality, 12 cases. Now, these are not risks. These are disproportionalities from a particular database. It gives the risk of finding this among TKIs, finding this cardiotoxicity in that drug instead of all these other drugs that happen to be in that database. Some people, by the way, do disproportionality. Say people work for pharma company ABC, and they do it among all the drugs in their database, and that's highly biased by the drugs that company has developed. But a risk is how many cases per 100 patient years. We must have that exposure as part of risk. Then we take the risk ratio of the experimental drug or the drug of interest to the control group. And that is a ratio of 2 Poisson variates. We know how to handle that. We can use Poisson regression. We know how to compute a confidence interval. And we can even call that ratio a hazard ratio. I sometimes call it a poor man's hazard ratio. It doesn't come from Cox's model, but it's a good start and a good estimate. That is the way we do it. That's the way we're used to doing it. And there are these concerns. And Michael, you might have some concerns about people reading the paper that I just described.
Yeah, and I think it's important. What oncologists want to do is they want to maximize tumor response and prolong meaningful life. I think that if you were to survey oncologists and say, what are you trying to do? Well, we want to eliminate the tumor, and when we can't eliminate the tumor, we want to maximize the tumor destruction with the intent of prolonging meaningful life. If you ask the cardiologist who deals with oncocardiology, what they want to do, or their primary concern very often, probably not exclusively, but very often, is they want to minimize the cardiac effects and preserve cardiac reserves. By preserving the cardiac reserves, patients who are now living longer because of better cancer care will have less late complications. So their goal is to minimize the cardiac effects. Now, ideally, these goals should be congruous. We should say, wait a minute, if we can manage to maximize meaningful life, we may have to accept some cardiac risks. And so the goals ideally are congruous, but they're really kind of have evolved in somewhat separate ways. And so the idea of minimizing cardiac problems can sometimes interfere with ideal cancer treatment. You asked about the paper. Well, I think the paper is useful in raising questions about the use of TKIs, but certainly further investigation is necessary. We can think about some instances where fear of a cardiac event has interrupted meaningful therapy. What happens? Well, if the oncologist suspects a problem or is advised that there may be a problem, they can sometimes adjust the dosage and say, well, we better be careful, but adjusting the dosage from what the oncologist thinks is ideal could be detrimental. We could add surveillance points. Okay, well, if we're worried about a cardiac event, let's monitor more often. But we've spent a good portion of the time talking about the fact that some of these surveillance points have false positives as well as false negatives. They're not perfect. Maybe from the oncologist's point of view, we should be really worried about this and maybe alter the therapy. We have other choices that we could use. We started out using the one we thought was best, but maybe we should use something that's potentially less problematic, might be less effective, and that certainly can be to the detriment of our patients. So this paper does not answer the questions, but it could lead to sometimes unfortunate changes in a clinical practice that could result in not achieving the goal of maximizing the useful longevity of a patient in the extreme way. Back to you, Jay.
Sure. Now, perhaps what we need are more post-market RCTs, and comparative effectiveness studies are becoming very important these days, especially being demanded by
Rosario, please mute.
Some consumer groups. What comparative effectiveness studies just takes almost all comers, and they are comparing effectiveness in the real world, but they will have the safety data as well. And so we used to call these large simple trials, and they are now being funded, and that may be better, but will industry sponsor these? What is, for any given pharma company, what is the benefit to them to have such a trial when they don't know what the results will be and what the heck, they already had their drug approved? But lack of these types of studies incentivizes people to do research on the existing databases. And that is where we get into the issue we are discussing today. Now, in an RCT, we can refine the AE risks. We know how to do meta-analysis of several trials and random effects models on individual patient data. I don't like aggregate data in meta-analysis, and I think most people are moving away from that now. We have the Early Breast Cancer Trialist Collaborative. This has been going on for decades. It is headed by Sir Richard Peto, but when he started this, he was not Sir Richard. That's how long ago this was. And several large institutions around the world pool their breast cancer clinical trial data. Every 5 years, there's a report on the progress that we are making, and it includes safety as well as efficacy. This would all be useful. Now, is there an analogy to meta-analysis for real-world data? We don't have covariates. We may have several sporadic reports of a specified AE type, say congestive heart disease, and maybe one comes from the FDA database, one from the Blue Cross Blue Shield, and some from Medicare. Can we combine these? I would think, and again, what are these, the data we're combining? They're most likely the disproportionality data that we the disproportionality estimates that we've already said are not desirable, but they might be percent of all patients or something like that. We might apply the James Stein shrinkage estimator or its close cousin, ridge regression, to at least pull these rates toward the overall mean, under the justification that we have only one observed instance of a repeatable event, and what that event is in real world data, we're not sure. But at least if we shrink the data, take the median and the interquartile range, and that may be something more meaningful to mull over than the report of any one database. But again, the interpretation may not be clear, but it does open up discussion for further research. Now, Herson and Robbins were concerned about this, and they were wondering, can we use data from these databases to emulate a target trial when there's not a randomized trial, when it's not available? In other words, let's say our ideal randomized clinical trial would be this, with these eligibility requirements, exclusion, etc. Follow-up, so on. Can we then go to the real world data and find people that meet an eligibility, a certain treatment strategy, etc. And in the follow-up period, all of these things that would be done. And at least this way, we'll have something that looks like a clinical trial, and even though it comes from these databases. Well, some people tried to do that. Harvard group, emulating randomized clinical trials with non-randomized real-world evidence studies. They have the RCT duplicate initiative, and they have started to see if they can follow the kind of thing that that Herson and Robbins were talking about. So they looked at 10 published trials, 8 with a cardio outcome, 8 were anti-diabetic trials, and 2 were anti-platelet trials, 7 placebo-controlled, 3 active-controlled. They emulated each trial with patients selected from medical claims databases, and they were not very successful. And we know they weren't successful with efficacy, and they weren't so successful from what we know so far with safety data either. So that brings us, is there any hope? Yes, some of you may have been thinking of record linkage as I taught. For each sporadic report from an insurance claims database, can we link to the patient's complete EHR, where there are data on comorbidities, concomitant meds, EKGs, etc. And there may be linked to a cancer registry where the patients are found, and then compare different treatments. We now have the tools that are developing of artificial intelligence, machine learning, natural language processing are being developed as we speak, and these can also be used to bring the data together in a more useful way. FDA is participating in Project OneSource. This is a continuous, efficient transfer of EHR data to a common research format. That is, wherever patients were treated, now this is their EHR data. I'll tell you how the sporadic gets into this, but now researchers have this rich, the database is the same, regardless of whether the patient was treated at MD Anderson or UCLA or Vanderbilt. Now, we can now link, now going back to record linkage, link the sporadic report into this Project OneSource. However, we may find the data are already there because the patient visited their doctor and it got onto their regular record. And that's good. We can remove duplicates in that way. All of this is coming in the next few years. The Project OneSource just got underway. Now, FDA has had the Sentinel initiative for a long time, pooling of insurance claims data, EHR data with tools for analysts. It's not used that much. There are problems with it still, but this initiative is being improved with the Project OneSource, as far as I can see. But we're talking about the US. What about the UK National Health Service? The UK has sporadic reports as part of the cradle to grave records of all of the king's subjects. That we could be doing analysis in Britain. And in fact, Scotland just did an analysis of long-term COVID. And I go on the metro and I see signs on the walls of the metro here. NIH is looking for people between the ages of 6 and 12 years old to follow up for the rest of their life. If they had COVID, follow them up for the rest of their life to see about long-term COVID. But that's good. But the UK has these data, and they just reported just from their clinics in Scotland, an excellent report on long-term COVID. So we should be looking to our cousins across the sea, and if they can ever agree on a prime minister, that may help us use their data and maybe combine our data with theirs someday. Michael, do you have some final words, clinical?
Yes, and I think we're running out of time. Can we go 2 more minutes? Is that acceptable? And we'll try to finish up.
Of course.
Okay. So
we
as far
actually
as
have 32 more minutes.
Oh, okay. So we're
we
great.
still have plenty of time for the discussion.
So that's great. So final words from the clinician.
Michael, he has 30 more minutes.
Okay. Final clinical thoughts from physicians feel that some level of safety if they follow guidelines. So this is kind of a little bit apart from what we've been saying, but if we have guidelines, so people have looked at this data, maybe good data, maybe data that isn't so good. But if we have guidelines, physicians intuitively feel that they are on much safer grounds if they follow the guidelines. If something bad happens and they were following the guidelines that said they were doing what they should be doing, they feel safe. If something bad happened and they weren't following guidelines, then they have to defend what they were doing sometimes. And they have to come up with a reason why they didn't follow the guidelines in a particular case, and that can be very challenging. So why bring this up? And we bring this up, or I bring this up now, because as we create these guidelines, if we are really, really sure that they are good and they should be followed, then that's great. But sometimes expert opinions are wrong, and it can take a long time to figure out that they're wrong, and it can take a long time to make it right. So I kind of remind you of a recommendation from a while back. Perhaps we have some older people in the audience that remember. But mitral valve prolapse was all of a sudden a huge problem, and endocarditis was considered a problem. So why not give intravenous vancomycin before dental work for those who had mitral valve prolapse? Well, there were a couple of problems. First of all, mitral valve prolapse was overreported because the criteria was not very good. And secondly, the risk was not all that good and not really all that well studied. We overcalled prolapse, we overused antibiotics, and lots of clinicians followed the guidelines and thought they were doing the right thing. It's easier to write a flawed guideline, by the way, than it is to correct it. And we may be seeing some of these things in the relationships that Jay and I have tried to share with you. So be cautious of guidelines based on incomplete data and uncertain assumptions. And remember that expert opinion is an opinion, perhaps of many, but still it's just an opinion. And back to you, Jay, for some perhaps final words.
Yep, I think these are, as a statistician, I would say, let's not forget risk-benefit. The RCT is a useful construct for getting the drug approved, but its use in clinical practice is limited. Sporadic report databases are not adequate to categorize risk alone in post-market. Reports should not be ignored, but used with caution. We have imperfect estimates of risk, regardless of origin, RCT or RWD. Michael and I published this paper in Cardio-Oncology. It has most of the things, not all, but most of the things we discussed today. It was published in July in this journal. And we thank you.
Well, that was a great webinar. And I particularly appreciate how you guys were bouncing back and forth, working together in the presentation of your work, just like I imagine you work together, right? And in fact, what we're trying to emphasize with this webinar series is that you can't work together in parallel as good as you can dynamically, interactively working together. And since I'm the moderator, I get to ask the first question. And I always want to know about the engagement in the partnership. And you guys provide a very motivating example. And one thing I really want to emphasize here, this is not a simple problem. And you are both very accomplished in your own disciplines, right? So this is not collaborating together on a low level. This is a complicated problem, and you're working very dynamically together, which is historically not a strength of, I'm a statistician, so I'm going to say this from the statistician side, is not a strength of statisticians being able to collaborate with their medical colleagues. So I would like to ask each of you, I'll start with Jay again, because I'm a statistician, but then Mike, I also want to hear your perspective here. How have you been able to engage so effectively together?
Yeah, well, thank you, Greg, for observing that. It has been true of us, and I think Michael and I both know it.
Jay, could you stop screen sharing so we could see you better? Thank you.
Okay. Yeah, so I think Michael and I have both observed that this has been a fine collaboration, and it isn't over. I was looking this morning at some work we're starting now for still another paper. But I would go back to, I once worked in a cooperative oncology group when I was at MD Anderson, and the head of the group of oncologists said, I had made a comment, and he said, that is a clinical question. You have no business commenting on that. But what I knew then, and what I've learned even more, there are no clinical questions. There are no statistical questions. There are only scientific questions. And I think Michael and I both know that and observe that. And that's why it has worked out. I think our collaboration here on the screen is a little more calmer than some of our collaborations that we've had, but nevertheless, and I think there's a tremendous advantage from growing up in Queens. Mike, what would you say?
Well, I think the key to this is that we are both attempting to work out the best care for our patients on a scientific and not an emotional basis. And so we put all of these other things aside, and I'm not a statistician. I had the unfortunate experience in my public health training and to some extent in my undergraduate training of taking some statistical courses, but I found the most useful was to be able to look at data and say, this makes sense or this doesn't make sense. So I have the wonderful opportunity to approach somebody of Jay's status and say, Jay, I don't think this makes sense. How do we look at this problem? And Jay will be able to come back and say, well, this is the way we might analyze this. Does it make sense clinically? And we can banter over that, and we can agree or we can disagree. But in the end, we come up with something that both of us think works. And I think we can do that because we don't have one of us that leads. We have one that leads in some aspects and one that follows in some aspects. And we are absolutely, at least my impression, and we hadn't planned on this question, but my interpretation is that we are approaching these questions on absolutely equal footing and equal respect and equal acknowledgement of our own particular strengths and weaknesses to come up with something that works.
Right, and I would also add to that, that if I were at MD Anderson at the same time with Michael, and we were both employees of MD Anderson, I believe the collaboration would be the same as it is, as we are now part of independent institutions.
All right, that was very interesting. Moving on here, Karen Stein has a question about comparing real-world evidence to randomized controlled clinical trials. So if she wants to get on here to ask the question, but I would point out that a lot of times we talk about how randomized controlled clinical trials is the gold standard, but there's a lot of useful information, as you clearly elucidated in your presentation together. So Karen, do you want to ask your question?
I had actually several questions, and I'm wondering, maybe as a observer of everything I was writing, if you wanted to summarize them. I mean, I had questions about using the Poisson distribution. I had a question that we've discussed at length, the risks inherent in how we capture the risk portion of this equation, but we didn't at all discuss the inherent risks of trying to quantify benefit. And I felt like we get to this level of specificity on the risk half without necessarily making sure that we've done applied the same level of rigor to the benefit. I also feel that, at least for the US-based data, we do have some detection biases in that not all patients are insured, and that many patients cannot or do not seek health care for some of their cardiac symptomology until it gets severe, and that would create a bit of more of a detection bias. I'm happy to hear that the UK, which has a more thorough coverage, would be somewhat immune from this. But again, they're going to have a detection bias in the sense that the very wealthy often purchase private insurance and step outside the system, so their detection bias might go in the opposite direction. But I also want to hear again about the choice of Poisson. And then finally, I'm wondering, I know this is a lot, but I figured I'll just summarize everything at once. The concept of using case control or registry, which you mentioned the UK is doing, rather than some of these real world evidence, and whether that might help alleviate some of the biases. My last comment is that I am a proud graduate of the Hopkins MPH program. So thank you for that, your introduction.
Can I start with perhaps the first part of the question? Is that okay?
Yes.
Yeah.
I'll leave that to you.
And I'm going to take on a certain arrogance because I'm not the statistician. So I'm going to offer a mathematical answer to your question, and I'm perfectly comfortable about being shot down. But in many analyses, and Jay and I are actually working on this, we're looking at that balance between efficacy and risk. And so here is a thought, and perhaps some of you would like to comment on it. And that is, if we take the likelihood of risk and the severity of risk, okay, two factors, and we assign those a numerical value, and we multiply them together. So we then have the problems. On the other side of the equation, we have the burdens of eliminating those problems. And that burden can be cost, that burden can be ineffectual treatment, that burden can be inconvenience. And when those burdens exceed the product of what we are trying to eliminate, that is how severe it is and how often it occurs, when the burdens are less, we need to react. When the burdens are greater, we need to step back. How do you all feel about that mathematical approach to her first question?
You sound very Bayesian.
He is, believe me. Yeah, I think it's a good approach. I would say that to answer first what Karen was asking,
that's just the first of her questions.
Okay, yeah, I love the approach. Go ahead.
No, that's it for me. But I would be interested if anybody thinks this is crazy or not.
Well, it's not usually the language we hear from a clinical safety professional, but it is very nice that you have this language where you can discuss these ideas with your statistical colleagues.
The concept, by the way, originated from a famous legal case. And that's something that if anybody needs a reference to, but that type of analysis is certainly not new and certainly not original with me.
And Michael is very often bringing his legal training together with his clinical training as we talk. And I'm particularly interested in the legal startings of what he just said. But Karen, we were aware that we were not covering efficacy or benefit. We had to carve out a smaller part here, but we took the attitude that yes, trastuzumab is effective, it's approved, it's being used, it's being combined with several drugs and still is effective. But what about the safety? And we thought that was the weakness, and that's why we did that. For the Poisson, it's the rate per 100 patient years. It takes exposure into it. Mean per unit that in rare events is usually Poisson. We usually use that. And that's why I have always used that rather than just the binary variable of the percent of patients who had this, because in some groups, the patients have less exposure because they went off treatment because the treatment was not effective. But then again, oh, look at how many fewer adverse events they had. So I give an example in my book of that lung cancer. But anyway, I hope that answers your question.
Related to that, so you pointed out that real world evidence provides a different perspective and an important perspective. Now, you have these two different sources. You got randomized controlled clinical trials, you got real world evidence coming in. How do you combine them together? For example, for the healthcare provider, or maybe understanding results from a registry, which is kind of a hybrid of these two things. How do you use this information in a learning and decision-making process?
Michael, you want to go first?
I'm not sure that I'm the best person to answer that. Go ahead, Jay.
Yeah, as we pointed out, Greg, there are many, many problems in trying to combine them. But I do think they have to be given as two different measures. In the clinical program, we saw these cardio adverse events at this rate. And what we're seeing from databases is we're getting patients of this type, so many reports. And in many cases that people will say, well, this is in line with the clinical data, or this is more than the clinical data. On the other hand, it might be more than the clinical data because of false positives, because of the type of reporting, the kind of biases we have mentioned. We are combining them by discussing them separately and cautioning people on what each means, including that the RCT data had a limited patient pool and with limited inclusion criteria.
Very interesting. Do you think there's a potential for numerically combining them together into one estimate, or do you think that that's not advisable since the information is so different? Maybe it's better to use kind of a dashboard.
Yeah, well, I think with record linkage and Project OneSource, as I mentioned, we're going to be able to do that someday. But then we'll have all the data in one place with all the covariates. And, if the Blue Cross database has one claim and we bring that into their EHR, well, look at all the other data we know about that particular patient now that we wouldn't have from the claims data. That is the future.
The next question is from Heather Howe. If she wants to jump on here, I'll go ahead and read the question, and then she can expand on it if she wants. What is the appropriate interpretation of disproportionality results? This seems like a good method in a large database.
Well, what it means, again, it's how frequently the adverse event was observed in the drug of interest compared to how frequently it was observed among all of the other drugs, and what is the ratio of that? As we pointed out in that paper on the leukemia, 3-to-1, 4-to-1. It is a risk of finding it in this kind of database. It is a relative risk of finding it in this kind of database, but it's biased by the drugs that happen to be represented in the database. And as I said earlier, we have to realize that the drugs that are also in the database are there only because the patients had represented in those drugs had an adverse event of some kind, or they wouldn't be in the FDA database. Now, there are other databases like Medicare where they would be in, regardless of whether they had an adverse event or not. I hope that helps a little. It's hard for me to say anything tremendously positive about that disproportionality.
Yeah, I'd like to get your opinion of disproportionality analyses, Mike.
Disproportionality analysis to me as a non-statistician and a clinician is, if we look at something and we balance them, is this proportionate? And if it's not, call the statistician.
Yeah, it's a good,
find out why.
Right. It's a
find
good
out
signaling.
what are we missing clinically that the statistician can help us and can lead us down a path of finding the right answer. And that is really where the interaction is so important. And this is really going back to utilizing our resources. And if we have the resource to say, wait a minute, this is disproportionate, I don't understand it, help me. The person to help is very, very often the statistician that can look at that and can offer us different paths and the most likely path of an explanation.
That's a good explanation. I like that. And I think that is relevant for the new arena with machine learning and artificial intelligence, that these are mathematical tools that can help signal things, but then that doesn't give the final answer. That just alerts us that we need to have a dialogue about this and do a scientific evaluation of the signal. But moving on, because we're running out of time, Nancy Geller has the next question here. If a CA, I think that's cancer, if a cancer patient has an adverse cardiac event, surely the oncologist would make a dose modification. The problem arises from generalizing this dose modification to everyone with that cancer. I think that sounds more clinical. Maybe we can start with you, Mike.
Okay, the first question is the cardiac event real? And the assumption of the oncologist contains two possibilities that are unfortunate. First is that it's real, and the second is that it's related to the drug. Therefore, the oncologist will very often say, okay, we have to modify the treatment, either a lower dose or put in a rest period or use a different drug or whatever the oncologist then decides to do. The concept that Jay and I are trying to foster is, instead of that and the assumption that this is what we as oncologists then need to do, has to be, wait a minute, how do we balance these risks? And first of all, is it real or not real? Is it really related to the drug in question, or is it something else? And what are the likelihoods that the drug is responsible for this? And if the likelihood is extremely small and the risk of changing is very big, that has to be a different decision than if the likelihood is high and the risks are big. Then we absolutely need to do something, even if we're not 100% sure of why.
I think there's a tendency to err on the side of caution.
And what we want is to understand that and to maximize outcome by balancing these things better. And that really is perhaps indirectly, but very much the intent of the focus of what Jay and I have been doing.
Yeah, the problem is that clinicians tend to act quickly.
And sometimes to the detriment.
Absolutely,
of
absolutely. But
patients.
still, they err on the side of caution because just in case, they want to be sure they don't get a repeat event.
And they want to follow guidelines. And I talked a little bit about that.
Right.
Now, the best example that I can give is a little bit older, but in the early trastuzumab era, there was a great deal of concern about cardiotoxicity. And there was even one report that said, oh, there's 27%. Fortunately, that has never been duplicated. But a whole clinical trial arm was shut down. And the arms that stayed open demonstrated a very highly significant reduction in recurrent breast cancer. So in actuality, the drug statistically was curing the disease in a significant number of people, but was stopped because of a false concern of cardiac events. And these are very troubling things from the past. We want to eliminate those things in the future. And it is a hope of getting oncologists to understand not just that there's a cardiac moment, a cardiac event reported, but to put it in the perspective of, is it real? Is it serious? What is the effect? And what am I doing on the other side of that hypothetical equation that I mentioned before? What are the burdens of making a change? Is the change something where I have an equally effective alternative and it doesn't make that much difference? Or am I really harming the patient? Are you asking these questions from a statistical or a clinical perspective?
I'm a statistician, but some of my nearest and dearest are patients.
Yeah. Okay. Hopefully this
Thank you.
offers
It was
some
very
perspective.
interesting.
Yeah.
It was a very nice session. Thank you.
Thank you. And I think Nancy, I agree with what Nancy and
So let me just have a couple concluding remarks. There's one more question I'd like to throw out to give both Jay and Mike one last opportunity to say something. But I think this has been a good year. This is our second year with this webinar series. And we intend to do this again next year. We haven't started organizing yet, so we can't advertise right now. I do want to let Jay and Mike know that there's a lot of positive comments in here. They range from interdisciplinary collaboration is excellent to, the New York accent aids the collaboration. And there is a question about the slides. They will be made available, probably not today, but soon for everybody who is interested. So the last question here, and I'll let you, Jay and Mike, decide who goes first and who goes last here. But this really gets at the crux of the matter here between the partnership between clinical and statistical scientists. Seems subjectivity still is needed. Where would the weights come from? And this is from Kraylin Davis, if he wants to jump in too. But Jay and Mike, you can decide who speaks first and who speaks last.
I think this is actually referring to the hypothetical equation. So if that is correct, obviously all of those variables that I said should be multiplied with each other or compared are totally subjective at this time. You could give values to some things, but they would never really be quantitatively accurate because every patient is different. So in thinking about the subjectivity of that equation, let me put it in a perspective of this type, and that is, if you have an ejection fraction decline that almost always reverses, that's a very, very low likelihood. And subjectively, you can't say it's a number, but you can say it's small. Or you can say that this patient has developed clinical congestive heart failure. This is big. And so if you look at that relationship as a qualitative rather than a quantitative one, you can still say one side of that is so much greater than the other that we need to intervene or we need not to intervene, depending on which is greater.
Yeah, and I think it's a really good question. And our next paper, Michael spoke of the burden, as well as the frequency of adverse events and their severity and that inequality. We'll spell that out, but the burden is going to be a linear function of several factors, and they will be weighted. Now you're asking, where do the weights come from? What we think we would do is we just get this method out there, and then each clinician will assign weights of what he or she thinks is important for this patient in this instance or this group of patients. But we would not impose weights on anyone. Now, where will an individual physician get the weights from? From their practice and experience, as they do anyway in their mind without formally writing them down or computing inequality. So we'll see. We won't have a perfect answer to this, but we are looking into it.
The clinical criteria for the seriousness of adverse events is generally looked at a scale of 1 to 5, where 5 is death. And 1 is a concern that has not expressed itself clinically. So we might actually start with that type of a grading because it's so well established already. I want to take this opportunity to thank the organizers for this invitation. Also, as we just heard, we're running out of time here. But I have not just considered this a wonderful experience and a challenge, but I've also considered this a great joy to be able to interact with all of you and with Jay and the organizers. So I want to say a huge, big thank you for this wonderful opportunity.
And I thank the organizers too, and the help they gave us yesterday and in the dry run, and all of you, especially those of you who stuck around since the beginning.
Well, that's a great way to end it. And I really enjoyed it too. And I think there's a great value here. So thanks, Jay and Mike. And we hope to see our audience again next year. Look for advertisements. We'll have more partnerships to tell you about.
Any further questions can be sent to the organizers of the meeting. They'll be forwarded to us, and certainly I will be happy to respond.
And that would include the questions that were still in chat that we didn't get to answer.
Sure. All right. Well, thanks again. Great webinar. Have a good day, everybody.
Thank you all.